All right, so already working on the next video for photosynthesis because we need to test over this unit next week. So photosynthesis depends on chloroplasts in green plants. Also in any eukaryotic organisms, you're also going to have chloroplasts being the major photosynthetic organelles. So the pigment that, and excuse the crumbling, that is my cat deciding to walk on things. So the major pigment that chloroplasts use is chlorophyll. I don't know if this will help you remember anything better, but chloro is actually a prefix that means green. So very creative. They named them after the fact that they're green colored. Most chloroplasts are found in mesophyll, which is the interior kind of spongy looking tissue of a leaf. It's very spongy looking because there are lots of air spaces that will allow carbon dioxide to enter and oxygen to leave. Also, just a note on plant structure, they have these little they look like mouths, but they're pores opening on the undersides of leaves called stomata. Stomata are the main parts of a plant through which water evaporates, so they're the main way that water transpires from plants. That's the evaporation of water from plants. They're also the site of gas exchange, so they really do kind of do the same job as mouths, so that's why I call them little mouths. A single one is a stoma, more than one are stomata, and they are something that a plant can open and close to control air moving into and out of the leaf. Air needs to be able to make it to all of the cells in the mesophyll because those cells are performing photosynthesis, and for that they need a steady supply of carbon dioxide and they need to be able to release oxygen. The chlorophyll in a chloroplast is found in thylakoid membranes. So thylakoids are these interconnected stacks of these kind of membrane discs that you find inside of a chloroplast. They can be stacked up into these stacks called grana. A single one is a granum, but that's, only important just if they refer to it in a question and you need to know, oh, grana are stacks of thylakoids. So whatever I know about thylakoids applies to grana. Thylakoids are sitting inside a fluid called the stroma. These are the two locations we're going to focus on, thylakoid membrane and stroma, because that's where the major reactions of photosynthesis happen. So photosynthesis can be divided into two stages. The first step is the light reactions, also called the light-dependent reactions, and these are responsible for the photo part of photosynthesis. These require light in order to happen. So what they're going to do is they're going to capture light energy. They do a couple of tasks in the process. They are the reactions that actually use up water. Because remember, the equation for photosynthesis is CO2 plus H2O yields C6H12O6 plus O2. So the light reactions, let's see if I can highlight these. The light reactions are what use up water and what generate oxygen. The dark reactions, on the other hand, we'll do this in, how about purple? Oh, that is not what I meant to do. My bad. Apologies. Anyway, so CO2 plus H2O yields C6H12O6 plus O2. You should know this equation backwards and forwards. And I mean that literally because knowing this equation backwards is the chemical formula for cellular respiration, which we've already gone over. So you should know the equation both ways. So the light reactions are going to produce the water, they're going to break down the water and produce the oxygen. The dark reactions, on the other hand, let's make them blue, otherwise known as the Calvin cycle, are what use up carbon dioxide and what make sugar. During the light reactions, you're also going to be storing energy, so you're going to be making these high energy molecules like ATP and NADPH. And if you're like, wait, I know NAD, where did this P come from? We're going to talk about it in a little bit. The Calvin cycle is also called the dark reactions, also called carbon fixation, and those are the reactions that take energy that's stored in the light reactions and they actually build sugars. So they're going to take in CO2, they're going to add those carbons to a molecule to make a sugar. So let's look at a brief overview of where this is happening. So within the chloroplast, the light reactions happen on the thylakoid membrane. So you'll see water, NADP+, and ADP, and inorganic phosphate, are the reactants that go into the light reactions. And then what comes out is oxygen as a waste product, NADPH and ATP, both of which are storing energy. That energy is taken into the Calvin cycle, and the Calvin cycle takes in the energy from the ATP, the high energy electrons from the NADPH, as well as carbon dioxide, and uses that to produce sugar. Then it produces also as a byproduct ADP and phosphate, and NADP+. These are low energy forms of these molecules, which can then go back to the light reactions and pick up more energy. They kind of act like a ferry, like an Uber service, like a moving van, like a bus, and they carry energy back and forth between the light reactions and the Calvin cycle. So they go to the light reactions, pick up energy, take it to the Calvin cycle, their energy gets used, come back to the light reactions, pick up more energy, take it to the Calvin cycle, and drop off energy. And this just happens over and over and over again. So wow, that's bright. I hope that woke you up, because we're going to talk about the light reactions. The first thing obviously is going to be light entering the cell. What happens when light hits a molecule, if it hits an electron, the energy that's in that particle of light, which is a photon, gets absorbed by the electron, and then it enters what's called an excited state. This is kind of representing, this is, a y-axis with energy as its unit, and there's not really an x-axis. So as an electron absorbs a photon, it becomes excited, it has more energy. If there is nothing else around, if there's nothing to absorb the energy or to accept that electron, then what usually happens is that electron then releases a photon and then falls back to its ground state, or its less energetic state. That's what we witnessed when I was shining ultraviolet light onto chlorophyll, that chlorophyll solution. The photon that was released from that was a red photon, so that's why red light was coming from that chlorophyll. But in plants, that energy is not going to just be released as a photon of light or as heat. Instead, pigments are arranged into these reaction centers. Reaction centers are then surrounded by what are called light harvesting complexes, which basically contain more copies of the pigment. In plants, a lot of times this is going to be chlorophyll A. Together, the light harvesting complexes and the reaction centers are called a photosystem. And the very first photosystem that's used in photosynthesis is kind of oxymoronically named photosystem 2, and I'll talk about why in a sec. So all of this is happening in the thylakoid membrane, and what's going to happen is at the very end of this, you have a very special pair of chlorophyll molecules who receive all the energy that's being absorbed by this light harvesting complex, and that excites one of their electrons, and it's in an excited state, and it is then given to this molecule, which is the primary electron acceptor. It's going to accept that electron, and then it's going to go basically into an electron transport chain. So what happens is chlorophyll absorbs light, becomes excited. It returns to a ground state, releasing an excited electron. It's now got a positive charge because it's given up one of its electrons. So it's going to grab an electron from water, and in the process, it's going to split that water into hydrogens, some electrons, and also oxygen. And then in the process, you're going to form an oxygen molecule, and that's where the waste product is. So the purpose of water in this is just to serve as a source of electrons. The purpose of oxygen is it is a waste product left when you strip the electrons off of that water molecule. So once again, we're looking at this in a slightly different way. Photon of light hits a pigment, energy gets passed around until it hits that special pair of chlorophyll A, and then that chlorophyll A molecule is going to give an excited electron to the primary electron acceptor, at which point you're going to have some enzymes split water. They're going to produce some hydrogen ions. They're going to produce a waste product of oxygen, and they're also going to produce some electrons that are going to be used to replace the electrons that the reaction center chlorophyll have given up. O2 is a waste product that gets released. Then, you hopefully guessed it, the electrons that have been given to the primary electron acceptor are going to then go into an electron transport chain. And then the electron transport chain, just like in cellular respiration, is used to create a proton gradient. By the way, another name for that is the proton motive force. In this case, motive comes from the root word meaning motile, which means movable. So it's proton moving force. The force of protons moving is going to supply energy. And then we have ATP synthase using that proton gradient to create ATP. So, like I said, this all starts in photosystem 2, and then basically the electron's going to be passed along to photosystem 1. Why are they named backwards? Well, honestly, they were named in the order in which they were discovered. So when scientists were studying chloroplasts, they found photosystem 1 first, gave it a name. Then they found photosystem 2 and realized, photosystem 2 technically comes first, but by then the names had stuck, so they just stayed with them. So photosystem 1 takes in the electron from photosystem 2, sent down the electron transport chain. Keep in mind, a lot of its energy has been used up at this point. So the photosystem 1 absorbs more light energy and is going to give that energy to this low energy electron, which is then given to the electron carrier NADPH. And then there's also potential for another electron transport chain. So what's with the P? Well, it is simply noting that NADP is NAD with an extra phosphate group attached. The major difference is that NADP and NADPH are used in anabolic reactions. NAD plus and NADH are used in catabolic reactions. And if you remember, catabolic reactions break stuff down. Remember I said to think about cats and how they're very destructive. Yes, I'm talking about you. And then anabolic reactions are used to build. Like if you think about it, anabolic steroids are taken by bodybuilders to build muscle. So remember the difference between catabolic and anabolic. Those are things that you should not have to think about too hard. They should be memorized. All right. So now we have a diagram with photosystem 2 and photosystem 1. So we have this energy deficient electron that's been passed through the electron transport chain. It is going to then be donated to this pair of chlorophyll molecules, which we don't really need to talk about the P680 and the P700. We're just going to call them P700 so I can quit saying pair of chlorophyll molecules. When light hits this photosystem, the energy is going to be passed along from chlorophyll to chlorophyll until it hits that P700 reaction center. It's going to boost that electron to a higher energy level, and then that is going to be given to the primary electron acceptor. From there, it's going to go into another electron transport chain, a much shorter one, and it is going to then be transferred to a NADP plus, converting it to an NADPH, which now has a high energy electron that can be used in the Calvin cycle. A lot of stuff going on. You do not need to know the names of any of the molecules in the electron transport chain, either between photosystem 2 and photosystem 1, or between photosystem 1 and NADP plus reductase, which is the molecule that makes NADPH. You don't even need to know the name of that. Most important thing would be to recognize, hey, it's got ACE at the end. It's got to be an enzyme. So this is just an analogy that some people find kind of useful to show the energy and the work that's being done by these photosystems. So photosystem 2 is where it starts. A photon comes in and it gives energy to this electron that's represented by the height of this electron. So up here it's a high energy electron because it has in that spot high potential energy. It is then used, some of its energy is used to make ATP, at which point it enters photosystem 1. Another photon enters, gives it some energy, so it's now in a high energy state, and that energy is used to make NADPH, which can then use that high energy electron to do some work. The electron transport chain is very similar to the electron transport chain that you see in cell respiration, in that you have molecules that are passing an electron along, and as they pass the electron along, they use up some of its energy to pump protons across a membrane. So you're going to build up a high proton concentration in the thylakoid interior, which, if you're being fancy about it, is the lumen. And then as the hydrogens diffuse out into the stroma, remember I said we were going to talk about about thylakoids and stromas. As it diffuses out, ATP synthase uses that movement of the hydrogen ions to produce ATP. This process is called photophosphorylation, whereas in cellular respiration it was called oxidative phosphorylation. The difference is that photophosphorylation has that photo in it, which reflects the fact that light is involved in this process. Now we get to the Doock reactions, otherwise known as the Calvin cycle, otherwise known as carbon fixation. So this is step 2 in photosynthesis, where the energy that was stored in the ATP that we made and the NADPHs that we made is used to, quote, fix carbon dioxide into carbohydrates. When we're fixing carbon, that means we're basically taking carbon from carbon dioxide and inorganic molecules and attaching it or affixing it to an organic molecule, and that's how we make carbohydrates. This process happens in the stroma. It's a series of reactions and like in a cycle, just like the Krebs cycle or citric acid cycle was. Each of the steps of it is going to be catalyzed by a different enzyme. You do not need to know the names of most of them. And it can be broken down into 3 different processes. The first of which is carbon fixation. Then you're forming this molecule called G3P. And then you're forming basically the carbon acceptor that you start with all over again. So let's look at this in more detail. The only enzyme you really need to know about is Rubisco. And that's just because Rubisco may be referred to on the test, and it helps to know that it's basically the first enzyme in the Calvin cycle. So it is what adds in carbon dioxide into the Calvin cycle. It performs carbon fixation. It adds in a carbon. Rubisco is actually an acronym. That's why the name doesn't end in ACE. It's ribulose bisphosphate carboxylase / oxygenase, which is quite a mouthful, so they shortened it to Rubisco. This creates a 6-carbon molecule, which is super unstable, so it immediately breaks down into 3PG. You don't need to know what 3PG is. You don't need to know what its structure is. All you need to know is it's an intermediate in the Calvin cycle. I don't even know if you need to recognize it if you see it. Next step, it is changed to G3P, which is only important because that is exported to the cytosol and used to make sugars. So the G3P that's made in the previous step is a 3-carbon sugar that is used to then make glucose, fructose, and any number of other 6-carbon sugars. So the source of the glucose that's made at the end of photosynthesis is basically this stuff. So you have to produce 2 G3Ps, since they only have 3 carbons each, to make 1 glucose. So the very end of this, you need to reset the cycle and get everything ready to accept another carbon dioxide, and that's regenerating this carbon dioxide acceptor. It's called RuBP. You don't really need to know much about it. Just need to know that there's energy required to produce it, so that way it's able to accept another carbon dioxide. And this cycle just keeps going on and on as long as there's energy to power it. So let's take a look at it. Once again, you don't need to know the names of molecules. Most important one to know maybe is Rubisco. Maybe that G3P at the end. So step 1, you're fixing carbon. You're adding in another carbon dioxide, another carbon from carbon dioxide. We're just going to think about this as 1 carbon dioxide entering at a time. So it's going to end up becoming part of these 2 3-carbon molecules that are formed, the 3 PGs, don't need to know what they're called. Next step is going to be using energy from ATP and using NADPH to provide high energy electrons, and that is a phase called reduction. It produces G3P, this 3-carbon sugar that's used as the base for making a lot of different organic compounds. Now, you cannot just leave this process stuck here, because if you don't reset this cycle and produce the RuBP, you will not be able to continue the Calvin cycle, and you won't be able to accept more carbon dioxide and build more sugar. So the last step is to regenerate that RuBP, just so that way the cycle is reset and ready to go again. Just wanted to point out a few things in comparison between photosynthesis and cell respiration. You do use chemiosmosis, meaning using that proton gradient to generate ATP in both chloroplasts and in mitochondria. The major differences are in mitochondria, you're transferring chemical energy from food to ATP, so you're just going from one type of chemical energy to another, whereas in chloroplasts, you're transforming light energy into chemical energy. In mitochondria, you're pumping protons into the intermembrane space, and then ATP synthase is going to be powered by the hydrogens diffusing back into the matrix. In chloroplasts, the protons are pumped into the thylakoid space, which drives ATP synthesis as it diffuses back into the stroma. Here's just another diagram that I thought was pretty useful, showing you, one, the locations of these things, and showing you how chemiosmosis is basically taking the same path in both of these molecules. There are differences in that there are different molecules in the electron transport chain of each of these processes. They, take place in different organelles, but the ultimate properties of it, the ultimate series of steps in the process is the same. And then I just wanted to go back to the relationship between photosynthesis and cell respiration. Remember, cell respiration is performed by all organisms. If you are alive, you perform cell respiration in some format. The common type that we talk about requires oxygen. Then its waste product is CO2, which is one of the reactants in photosynthesis. Photosynthesis is only performed by producers, plants, algae, and certain bacteria. Now, keep in mind, cell respiration is going to make ATP. That's used to power things that are happening in the cell. Whereas photosynthesis has an input of light energy, that ATP is then used to make sugar. I want to point something out. Plants and other photosynthesizers perform photosynthesis and cell respiration. They do both. They make sugar, which is how they take in the light energy and store it long term, but then they have to use that sugar in order to power their life processes. So generally, producers will produce by photosynthesis a lot more glucose, a lot more chemical energy than they need to consume. And that excess will get stored as starches or some other storage molecule. If you were to take a plant and put it in the dark, so that it could not perform photosynthesis, it could use up those stores of glucose for a little bit to power cell respiration. But once you use up all of the glucose, all of the carbohydrates that are stored in the plant, unless you let it enter light again, it will then use up all of its energy source and it will starve. So you may see questions on AP tests about differences between having chloroplasts or other photosynthesizers in dark conditions versus light conditions. What's going to be the difference? What gases will be produced? What processes will be performed? So all of that is something to keep in mind, and to remember that plants do photosynthesis and cell respiration both. So they both consume oxygen and carbon dioxide. The main thing is they produce more oxygen than they consume. So that extra oxygen gets put into the air, and that's what we breathe in. All right, that is the end of this lesson. I know it's probably confusing. Maybe watch it a couple of times. You can watch some other videos online to try and help you understand this process. Like I said, complex.